Thank you, Ed, for the introduction. I'm Yue and Erin, and I'm really glad to share our work today with you. Neurons endure many forms of stress when establishing and maintaining their function. Disruption from intracellular or external environment triggers neuronal stress sensors. It transduces retrograde signal to initiate transcription events and lead to either neuroprotective or degenerative processes. So how is the stress sensing signaling regulated in neurons? In this collaborative project, we combine C. elegans and mouse model to dissect the molecular mechanisms that control neuronal stress signaling. Our studies focus on the conserved MAP triple kinase DLK family. It includes the worm member DLK1 and two mouse members, DLK and LZK. DLKs act through the MAP kinase cascade to induce transcription factors. Loss of DLKs causes axon unable to repair after the acute injury and slows down neuron degeneration. So to understand the regulation of DLKs, we use forward genetic approaches in C. elegans to identify the neuronal regulators of DLK1. And in mouse, we apply transcriptomic approaches to dissect the cell type dependent gene network that underlies the DLK-initiated neuron degeneration. In C. elegans, I first visualized endogenous DLK1 by GFP tagging. And then I did visual genetic screen to look for mutants that show increased DLK1, representing the context that activates it. From the screen, I found DLK1 being tightly controlled in the ciliated sensory neurons. These neurons have the long dendrites extending to the tip of head where the cilia sense environmental changes to drive the sensory behavior. The worm cilia neurons are conserved with the mammalian olfactory neurons. So in Y-type animals, sensory neurons form this fine structure at the dendritic ends, such as the wing shape in AWC neurons. And you can hardly see DLK1. But in the mutants that disrupt the ciliary transport, the structure is abolished. DLK accumulates at the tip of cilia. DLK misaccumulation at the sensory cilia has not been reported before, so we asked, what is the consequence? To examine the signal output, I looked at the transcription factor CEBP1, which is a downstream of DLK1 signaling. It does increase, and removing DLK1 restores CEBP1 level back to the Y type, suggesting the increase is dependent on DLK1. Active DLK1 signaling affect neuron function in many ways. So how do cilia neurons control it to maintain their function? Surprisingly, we found that the output of DLK signaling represses the input. As you can see in the Y-type cilia, the loss of CEBP1 does not affect DLK1. But in impaired cilia, loss of CEBP1 increased DLK1 levels significantly. The repression suggests an active feedback control of DLK1 in cilia under stress through the active signaling. So compared to the normal cilia, when it gets impaired, CEBP1 increases and suppresses DLK1. We reason that it may help prevent or slow down cilia decay. What if we block this process? Indeed, in one mutant where cilia shows the progressive defects, when we take away CEBP1, DLK1 is further accumulated. Meanwhile, cilia degenerates faster. We are now testing if the enhanced degeneration is due to DLK1. So from my study in the worm sensory neurons, cilia integrity is a stress source of DLK1. The negative feedback control of DLK1 level prevents cilia from degeneration, indicating the importance of controlling DLKs to protect neurons under stress. And now Erin will be sharing her study in mouse.
And so also be sharing my work using a mouse model to dissect the DLK-dependent gene regulatory network and what happens when you have too much DLK signaling. Oh. So loss of DLK actually slows down neurodegeneration in mouse models of disease. And under normal conditions, DLK doesn't seem to be active. So to study the active form of DLK, we actually used a transgenic model to selectively increase DLK in a Cre-dependent manner because increased expression of DLK leads to its activity. So I used mice to vGlut1 pre-mice to specifically increase levels of DLK in glutamatergic neurons. And I focused my analysis on the hippocampus. So here I'm showing RNA for vGlut1 and DLK at P15 or around 2 weeks of age. And you can see in that control mice, DLK is normally expressed in glutamatergic neurons. And I find approximately twice as much DLK in these neurons which have elevated DLK. So next I wanted to confirm that the increase of DLK activity in fact leads to, or increase of DLK expression leads to increased DLK activity. So in mice, DLK is part of a MAP kinase signaling cascade shown here, ultimately resulting in the phosphorylation of the transcription factor C gene. So to validate that the overexpressed DLK acts as a functional kinase, I stained for phosphorylation of the downstream transcription factor C gene. And we find that control animals showed baseline level of phosphorylated C gene. And then DLK overexpression actually leads to an increase in phosphorylated C gene, both in CA1 and CA3 pyramidal cell layers. So since I can confirm that the transgenes are functional, I next wanted to ask how the increased expression and activity of DLK affects these neurons. So to understand the consequences of increased DLK activity, I stained for nu-N, which marks neuronal nuclei at 4 months of age. And mice with increased DLK have glutamatergic neuron death. You can see here that the CA1 pyramidal cell layer is much thinner with fewer neurons. And we actually see degeneration beginning around P15. So together with previously published data, it seems that getting rid of DLK slows down neurodegeneration under various stresses, while increasing DLK promotes degeneration. And since DLK leads to the activation of the transcription factor C gene, next I wanted to ask how the increased expression of DLK affects gene expression. So to understand the DLK-dependent gene regulatory network, I've taken a multi-omics approach. I've isolated and sequenced RNA from the hippocampus and cortex at the P15 time point before most of these neurons die. And when I compare the expression of RNA between wild type and samples with increased DLK, I find 341 genes differentially expressed between the two conditions. I find that DLK activity leads to the upregulation of genes involved in stress response, regulation of neuron death, cytoskeletal organization. We also find strong changes in genes related to glial responses. So to sort out the neuronal and the non-neuronal changes in expression, I'm in the process of looking at the population of actively translated transcripts, specifically in neurons, as well as visualizing mRNA to look at which cell types exhibit differential expression. As a validation of my RNA-seq data, at the mRNA level, I've confirmed that the increased DLK activity leads to increased staphin-4 in neurons. And staphin-4 is a microtubule binding protein involved in microtubule dynamics. And this suggests that DLK may regulate the neuronal cytoskeleton through staphin-4. And just as an aside, the closely related staphin-2 has been implicated in ALS. And this is, again, a closely related gene. We also see among the top upregulated genes are several that have been linked to inhibition of NF-kappa B. NF-kappa B is a transcription factor widely known for roles in inflammation. And in the nervous system, it plays roles in hippocampal development and memory. So I'm currently investigating potential neuroprotective signaling through NF-kappa B.
You're at 9 minutes right now, Erin.
All right. So going back to what we know and what we've learned about DLK, DLK is a conserved stress response protein whose activity is tightly regulated. From Yue's work, we find that DLK senses ciliary stress in neurons and leads to neuroprotective signaling under some conditions, though too much DLK can be detrimental to the neuron. My work reiterates the importance of controlling the level of DLK signaling in neurons, with DLK regulating expression of the cytoskeletal gene STAFN4, and too much activity leading to death in the excitatory neurons of the hippocampus. So DLK acts as this double-edged sword, with some level of DLK leading to neuroprotective responses, such as regeneration following axon injury, while too much DLK leads to cell death. And coming back to our title about how to protect neuronal function during stress, we find that controlling the level of DLK is important for promoting neuron survival to stress. Neuron death is a common feature of neurodegenerative diseases, which often have unknown causes. So understanding DLK signaling will help identify therapeutics to promote neuroprotection. Our future studies aim to address the consequences of DLK1 misregulation in cilia degeneration, as well as identify changes to the neuronal translatome and possible neuroprotective signaling through NF-kappa B. So with that, I'll just end by thanking our collaborators who's helped with these studies, Stefano Brigidi, Brenda Bloodgood, Ben Hai Zhang in his lab, and also our mentor Yishi Jin, along with all the Jin and Chisholm lab members, including those who've helped me with the mouse work, including several undergrads who are not pictured here. And then last, I'd just like to thank the Kavli Institute for funding and providing this opportunity to share our work. So with that, we'll take any questions.
Great job, you and Erin. So there's a question in the chat from Nick, and he asked, is STMN4 upregulated also in C. elegans?
We have not checked if the homolog in the C. elegans is regulated.
Thanks. And while we're waiting to see if there are more questions in the chat, I had a more general question. So, I mean, I generally think about neurodegenerative disorders like Parkinson's, Alzheimer's, things like that. Are there reasons to think there are particular neurodegenerative disorders that DLK might be involved in and that these studies might have implications for?
So some studies have shown that phosphorylated CJUN, which is again the downstream transcription factor, but is increased in patients with ALS and Alzheimer's disease. So DLK could be contributing to that increase, but DLK is also a stress sensor for various stresses. So it could be potentially involved in other sorts of degeneration as well.
Cool. Thanks. Other questions? Terry has a question. He asks, what stimulates DLK expression in the hippocampus under normal conditions?
So under normal conditions, DLK doesn't generally seem to be active. It seems to be a stress response. So treatment with canic acid can lead to DLK-dependent cell death. I'm not sure of other stresses, but.
Now we're starting to get more questions than we're going to be able to get to. Next question is from Arti Narang. Is there a link between DLK and inflammation?
So DLK seems to, in glial cell, like in astrocytes, it can promote reactivity when it's active in astrocytes, and then other studies have suggested that DLK in neurons may regulate an immune response, though that's still, I think, being investigated. But DLK does seem to have an effect on the glial reactivity.
Great. We're right at 15 minutes now. So thank you and Erin for